Everybody, welcome back. This is the first of a few mini lessons on Chapter 5. I will be posting a lot more of these personally taught lessons for Chapter 5. I feel Chapter 5 is a pretty important topic. And so in addition to the other resources I've posted, I'm also going to be doing a little bit more lectures for Chapter 5. So at this point, you should have gone through successfully Chapter 4, which is kind of general cell structure and function. And then also some of the topics from Chapter 3, the lipids section in Chapter 3 is going to be very valuable. So if you weren't quite comfortable with the Chapter 3 material regarding lipids, you might want to go back and rewatch some of those videos on organic molecules or reread that section of the textbook, because we're going to be applying some of those properties of lipids to biological membranes. This first little mini lecture is going to be covering properties of phospholipids. We're going to review a little bit about what that is and then kind of see how they go together to create this phospholipid bilayer, which is what all biological membranes are made of. Let's take a look. So here we have just a lot of different pictures about phospholipids, but I wanted to just recall what a phospholipid is. So I'm just going to draw it right over here. So if you remember, we kind of have this general pattern of a phospholipid. It looks like a balloon with two tails or something. So if you remember, this top part is charged. It's going to have some partial positives and partial negatives. There's some polar covalent bonds. There's some actual full-on ionic charges on some of those atoms on that phosphate group. There's phosphorus and oxygen and nitrogen. And so that makes that end of the molecule polar. Whereas the fatty acids are nonpolar, because if you remember, when you just have a hydrocarbon chain, carbon and hydrogen, both of those have the same electronegativities, and they're going to have nonpolar covalent bonds. So we don't have any charge on that end. So if you remember some of the words that we use, when you have a polar covalent bond or ionic bonds, you have, I don't know if I'm going to have enough room. So this is the hydrophilic. I'm just going to write that, the end part of that. My cat's playing over here. So the hydrophilic end is on the charged side of the phospholipid, and the hydrophobic end is on that nonpolar hydrocarbon tails. So when you get a couple of these, or in this picture over here with a beaker, if you were to just dump in a cup of phospholipids into a beaker of water, you might get some of these things arranged this way. So if you take a look here, we have what's called a monolayer. So all of the hydrophilic heads are sitting on the surface of the water because they are hydrophilic, they are water loving, and all the hydrophobic tails are hanging up and sticking up in the air. Those hydrophobic tails would prefer to stick up into the air than be pointing down into the water. They are repelled by the water. Oil and water don't mix, right? So those tails are like oil, they're fats. So that is one of the ways that phospholipids can arrange themselves in water. Another way is what we call a micelle. So micelle, and here's another example of a micelle kind of in this other older picture, and here's kind of another one down here. It's called detergent, but it's similar to a micelle of phospholipids. So all of the fatty acid tails are pointing towards themselves because they're okay to be next to each other, with all the hydrophilic heads pointing on the outside because they're okay to be around water, but the tails are repelling any water. So they kind of push out all the water, and so just all the little tails are together inside the micelle. All right, another structure that can be formed. Again, this is just random, just the properties of these chemicals, reactions with themselves in water, is what's called a liposome or a vesicle. Liposome is kind of an older term. You can see this is an older picture. We now kind of call them vesicles, but the idea is now we get two layers of phospholipids, and we can have water inside this vesicle. So water is going to be inside next to the hydrophilic heads, and we have water outside. And so what we end up with is now we have a compartment, a separate compartment away from the watery environment. We can have a separate watery environment inside, bounded by this phospholipid bilayer. And then lastly, what's shown here is the bilayer sheets, and we have a bilayer here. So anytime you hear the word a phospholipid bilayer, that's what it's talking about, is we have a layer of phospholipids here and a layer of phospholipids here. Their hydrophobic tails are interacting, and the hydrophilic heads are pointing towards watery environments. That is the basic structure of plasma membranes, biological membranes, whether it's the cell membrane at the outermost part of the cell, like you learned in chapter 4, or whether these bilayers are making up the nuclear envelope, the endoplasmic reticulum, the Golgi body, the vesicles, the lysosomes, the peroxisomes, the mitochondria, the chloroplasts, anything that is made up of this lipid bilayer. This is the basic characteristic with some other stuff shoved in there. We'll see that in another mini lecture, but that's the nature of a phospholipid bilayer. And one of the reasons, the term that goes along with why this works, is this word amphipathic right here. All that means is you have a molecule that has both hydrophobic and hydrophilic parts to it. So a phospholipid is a perfect example of an amphipathic molecule, having a hydrophobic tails, the hydrophobic tails, and the hydrophilic heads. Okay, so that's the nature of phospholipids, and when they react in watery environments, bilayers are just a normal outcome of that reaction, and vesicles are a normal outcome of that reaction. Okay. So when we take a look at the variability in these membranes with the phospholipids, we can take a look at just some of their characteristics once they do produce that lipid bilayer. So in this, this guy right here, I'm going to talk about this diagram first. So if you were to be able to mark or tag or put like a little label on your phospholipids, so you can see they've done a green one and a purple one and a blue one, we would see that on that layer of the phospholipid, just this upper layer right here, that those membrane phospholipid molecules actually move around in their own layer very fast. So very fast lateral movement across a bilayer. They're saying once every 10 to the negative 7 seconds, which is a really short period of time. So that's like 10 millionths of a second. One move, they're happening like really, really fast. And that makes sense because they're not going where they don't fit. So the hydrophilic is still staying on the top and the hydrophobic tails are still staying pointing towards the middle. Now this example here, if we were to take a phospholipid and flip it over to the other side, that does not happen very often. So if you'll notice, it's very slow flip-flop hours or days. It might be assisted by proteins, but what that would be meaning is this hydrophilic, say the positive or the negative side, would have to travel through the hydrophobic layer. And it's repelled because it's polar. The hydrophobic tails don't like polar things getting in their way. So be comfortable with the idea that the phospholipids move very rapidly on their same side. They move not very often flipping to the other side. All right. Some other things I want to talk about is the fluidity of these membranes. So if you remember, again, you can go back to the chapter 3 lipids section. When you have a hydrocarbon, so this is just an example of a hydrocarbon. Every corner is a carbon, and you'd have oxygens, or hydrogen. So this would be a carbon, 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 carbon. Every corner is a carbon, right? Those lazy chemists like to do shorthand. And then coming off of every carbon, you're going to be saturated with hydrogens. Right? So that was the difference between saturated and unsaturated fatty acids, is when you're saturated, it's all single bonds between all of the carbons. When you had things like this, let's see if I can, I don't know if I did that right, but whenever you have a double bond, it's going to make a kink in that chain. So you can go back and see the difference between saturated and unsaturated fatty acids. And so that's what they're showing here in these pictures on the left. So when you have a whole bunch of saturated fatty acids, you'll notice those fatty acid chains are pretty straight. And if you remember, the difference is kind of in a large scale, kind of more applicable way. When you have saturated fats, they're solid at room temperature because those fatty acid tails can stack right next to each other. And so it's solid at room temperature, think animal fats. But when you have kinks, right, so this is where you're going to have those double bonds. You might have a saturated with an unsaturated. So here's a couple saturated and here's one unsaturated with a saturated. So you have those kinks. Then your neighboring phospholipids can't get as close to each other, and so that would be like our plant oils, vegetable oil and olive oils and those types of things, because those fatty acids have the little kinks in them because of the double bonds. And so when you have different environments, your cell has the ability to respond to those. That's one of the characteristics of a living thing. So if it's warmer outside, if the environment is warmer, they might make more saturated fatty acids to make it a little bit more rigid, because heat's going to loosen things up and we don't want our membrane to fall apart. If it gets too cold, we don't want them to be super close together, because then it might get too rigid and that could actually break. So then they might increase the fluidity, putting up a little bit more of those kinked fatty acid tails in a colder environment to keep it nice and flexible. So the cell has the ability to regulate which types of phospholipids get put up in the membrane in response to the environment. So I just think that's the coolest thing. And then lastly, another characteristic that affects fluidity is cholesterol. So again, going back to that lipids section in chapter 3, cholesterol is a type of a lipid. It is a four-ringed molecule, or four-ringed molecule. They make this the foundation for like steroid hormones and other types of neurotransmitters and chemical messengers. But they also serve an important part in membrane structure. So the more cholesterol you put into a membrane, and it's a lipid soluble, so it's going to hang out with those fatty acid tails, that cholesterol will make the membrane more rigid. The less cholesterol, the more flexible and more fluid it will be. So again, just like changing the kinks, straight or kinky lipid tails, the fatty acid chains, the cell can regulate how much cholesterol goes into or out of those fatty acids. All right, so that's talking about the fluidity once you have that bilayer. Okay, I had to pause for a second. My cat was being silly. All right, so this last slide for this particular mini lecture is just showing you, because of the properties of the phospholipids, they are chemically able to arrange with themselves. Remember, like we saw the monolayer, the bilayer, the vesicle. So here we have, we can actually read this both ways. Let's say we have some substance that the cell needs to get rid of. So these green circles down kind of inside of the cell. So this is inside. All right, so these might be, it could be a cellular secretion or proteins or waste product that the cell needs to get rid of. So it is in this direction that we're going to be seeing this vesicle come close to the surface. That bilayer of the vesicle will fuse with the bilayer of the membrane, and eventually you will get to release that product. So that's one of the ways that a cell can release its material, and this is called exocytosis. My cat. Of course, right, when I'm doing lecture. So that is one of the ways. We can also, if we wanted to, say, what if this was a cell, like an amoeba or something that's taking in substances? Maybe the green thing is food, or maybe it's a bacteria. It can kind of, the membrane is flexible enough to wrap around that substance and create a vesicle and then move that inside the cell. And then that can go and be processed by the Golgi, or the endoplasmic reticulum, or the lysosome, or whatever the cellular processing is of that. So this just shows you that flexibility of a vesicle and how it can fuse with the plasma membrane, or a vesicle can be formed by pinching the plasma membrane. So it can go both ways. All right, so that's just a little mini section on characteristics of phospholipids, properties of phospholipids, and how that phospholipid bilayer actually is made. All right, I will see you next time. Bye.